So welcome to this part of the talk. So today I'm going to talk about a couple of different things, and I decided to give a bit of an overview of our recent work on different diseases and some technological stuff, the autoencoders that we've been working on. And the way I picked these topics was I really tried to make sure for it to be in the spirit of the Pan-Cohort idea. So every one of those topics that I'm going to touch upon will have one angle or another on transferring data between different cohorts, using different cohorts for analysis, and so on. I also decided to stay pretty high level here because we have 25 minutes, and as you can see, a lot of different topics. So I'm not going to go as deep into the different papers. I'm going to show the data sets, basic ideas, and analysis we did. And then if you're interested more, you can read the papers or talk to me in person or virtually after. So the first one, which is not disease focused and sounds very technical, but has an interesting angle of cohort metabolomics analysis in it, is the project we recently published on variational autoencoders. Just to give you a brief introduction, the autoencoder is vaguely in the field of deep learning, although technically these networks are not even that deep, but it's this network-based type of machine learning model. And it can be compared to a PCA, principal component analysis, where you reduce your high-dimensional feature space, the metabolites in this case, into something lower dimensional that, intuitively speaking, gets rid of all the noisy stuff and keeps the good stuff. And then these components, similar to PCA that you have in that space, are supposed to be more meaningful than the raw data and show better associations. Now, these types of models, to train them, you need a big sample size, and at that time, at least for us, the biggest one we were able to access was the UK Biobank metabolomics data. So we had around 4,500 individuals there. And also, if you look at the population, they're heavily skewed towards female participants. So that will come up in a second. And we trained this model. And then you can look into the model. I'm not going to talk about that too much. So what are these components that you extract? Which pathways do they reflect? But the interesting part was that we then applied the same model to completely different metabolomics data sets. We had a data set from Qatar. As you can see, more balanced gender-wise, but completely different ethnicity. Then we had an AML study, a cancer study, American participants, and a schizophrenia study, again, mostly Qatari and slightly skewed towards the male side. So we used the autoencoder that has been learned on the twins data to encode these different data sets and then ran association analysis on that. And the surprising thing was, you can see, oh, this is not the cursor. Okay, I'm just going to talk about it. So what you can see is that the associations, the top points in those scatter plots, the strongest associations with the disease outcomes in the different cohorts were actually with the signal from the variational autoencoder, stronger than principal component analysis and stronger also than nonlinear principal component analysis. So these autoencoders see something in the data that transfers across cohorts that then correlates with diabetes or AML outcome here. And I thought this was fascinating. To be fair, this also decently works with the PCA itself, so you didn't need the autoencoder for that part. But the fact that you train something on a predominantly European female population and then transfer that model to another cohort that is completely different and still see that those components associate with disease was very interesting, I thought, and gives maybe also some hope for transferability across different cohorts. So now I'm going to go into different disease topics and cohort work that we have done there. I'm going to start with Alzheimer's disease. This is all in collaboration with our German partners and also Rima Kadura-Duk. I believe she's on the call. And there we work on different metabolomics and multi-omics cohorts in the context of Alzheimer's disease. I want to highlight one of them. There was a paper recently published, and it's specifically interesting because this is on brain data. Post-mortem human brain, about 500 samples, which is, of course, a difficult specimen to obtain, very precious samples. And then you see on the top right, we have various clinical parameters that were heavily discussed and scrutinized together with our clinical collaboration partners, especially David Bennett from Rush University, related to the pathology that these patients had in their brains, also cognition during lifetime and memory decline before death. And then you see that we have about half of the metabolites that are measured in the brain showing associations with these clinical outcomes. meaning that there's a massive metabolic dysregulation in the primary tissue of the disease. And then the rest of this paper goes through different ways of interpreting this and making sense of the data. The data is also public and also the code, so you can download all of this and reproduce these plots and do your own thing. I'm just going to show you a few things that we did here. First of all, again, in the spirit of the Pan-Cohort analysis, we ran some replication analysis, and anyone who's ever done any replication analysis, especially in metabolomics, knows how difficult this is. Different platforms, different panels of metabolites measured, so we could replicate about 30 of the hits, but we were also only able to see a subset of those hits because of different metabolomics platforms. Plus, these hits were in other brain tissues. So the original analysis was in the dorsolateral prefrontal cortex, and this is in different temporal regions of the brain. So, but those 30 hits that we can see across different brain regions were very strong hits for metabolic dysregulation that we believe are true throughout the brain. Just an overview, and again, you could draw 5 other plots like this based on this very rich data. These are just the pathways we picked for discussion that we thought were interesting. So you see changes in cholesterol metabolism. That's interesting because one of the main genetic risk factors for Alzheimer's disease is in a cholesterol-related gene. We see bioenergetic dysregulation, also related to mitochondria. It's an emerging topic coming up virtually everywhere. Some people say every disease is mitochondrial or energy-related. We see inflammation, so neuroinflammation generally has been a big topic, and we see the metabolic component of that in our data. And then on the left side of the circle, you see osmoregulation. That was an interesting sort of new thing that we saw. It's not really a pathway, but more a class of molecules that are related to water homeostasis in the cells. And there have been reports also in other neurodegenerative diseases that this osmoregulatory process, which affects both the cells and protein folding in the cells, as well as potentially the pressure in the skull as a restricted space, might make a difference in Alzheimer's disease. And we again see here the metabolic component of that regulated in different directions. Then one topic that we had in this data set, if you're from the Alzheimer's field, you're very aware of this. If you're not from the Alzheimer's field, you're probably also aware of this, is that there's this big debate whether the amyloid plaques, the protein that accumulates in the brain, which is part of the definition of Alzheimer's disease, is the causal factor for the disease. Very high profile drugs and very high profile discussions about these drugs, whether this works. And at least our piece of information here that we can add to this discussion is that, at least from a statistical point of view, so what we did is we looked at the metabolic correlations with amyloid and then the other protein, tau, and corrected the model for the respective other side. And it turns out if we look at amyloid and correct for tau, almost everything is gone. Those are the orange bars here. And if you look at tau and correct for amyloid, everything is already there. So it looks to us, from our data, that at least the metabolic component of dysregulation in the brain is much more strongly tau-driven than amyloid. And then as an outlook, just going to talk about this real quick. The research associate in my lab, Richa, she's still working on this, to now expand the analysis to really compare the differences in the different brain regions, because different brain regions are almost like different organs. There's completely different compositions and also functions of the brain. And we're currently looking with Mayo Clinic and other cohorts into how this distributes across these regions. One more comment. So now we're getting out of the brain into the blood. That's the easier specimen to work with. Of course, brain is not that easy to get to. So we have one interesting finding that I want to highlight in blood-based analysis. This is from the ATNI study, American Alzheimer's study. And so here we looked at the acylcarnitines, actually from the Bioenergetics kit, which tag mitochondrial metabolism, fatty acid oxidation, also amino acid metabolism in the mitochondria. And we, as many these days, looked at this concept of biological clocks. So we used the CORA data, the German data, to fit a clock model that predicts age from these acylcarnitines, these mitochondrial metabolites in blood, and then applied it to the Alzheimer's cohort. And what we're able to see, and this on the box plots on the right, is that you sometimes have clusters where, let's say, the ages, the actual chronological age, you see that in panel age is the same, so they have same age, but the biological age according to the model is much lower in one of those groups. You see that throughout this clustering tree, and it turns out that whenever the predicted biological age is lower, these people are actually better off in terms of disease. They have less Alzheimer's, better pathology. We sometimes refer to it as a resilience cluster. It could also be a resistance cluster. I'm not going to get into weeds of that discussion. But whatever it is observationally, the lower the predicted mitochondrial age, the better off also they are with respect to Alzheimer's disease. And we're currently still looking into how this goes further. And then to close the Alzheimer's part, Gabi mentioned this briefly, and I don't have a lot to show yet because the project is still pretty new. We're now starting to use this atlas, and I'm glad Gabi showed that in a previous presentation, this integrated network that they built and we helped build from many different studies incorporating pathways and statistical results to work on a drug repurposing idea, basically walking through these networks and finding unexpected links between drugs and Alzheimer's disease. And I have nothing to show yet. This is still work in progress. Okay, so we'll reset. This was Alzheimer's disease. Now I'm going to go to COVID. I think as many of us, we dragged into COVID projects and just did that, even though, I mean, me personally, I'm not an immunologist, I'm not an intensive care physician. So we worked here very closely with our clinical experts from Cornell. These cohorts that I'm going to present are all collected at the Cornell Hospital in New York, right in the beginning, March and April 2020. So this is actually multiple papers. Because we analyzed the data in different ways, and I'm going to show a little bit of everything, but we made this dedicated subpage on the lab webpage if you want to check them out. All data is also public, and again, codes, and you can reproduce everything if you go online and go to those papers. So just a few nuggets of findings here of what we saw. There was one cohort that was in the general wards of the hospital, so not intensive care. They were sick, so sick that they had to go to the hospital, but not so sick yet that they had to go to the ICU at that point when these samples were taken. Quite a big cohort, 230 patients. We had multiple time points, which made things rather harder than easier, but we used them in the model. And so what my back then PhD student came up with is he had all these clinical informations. Did they die? Were they on chemical ventilation? When were they released? So overall, how severe was the disease? So what you see in A, he came up with this composite model where we can put every patient on a scale from basically 0, the healthiest we have, to 330, the sickest person we had, by sort of a decision tree-like process. So, and then we have this ordinal scale of severity, and then he developed a mixed effect, because of the multiple time, mixed effect, ordinal, regularized regression model, and try to predict. And in B, you can see it's 70%, so we can debate about how much clinical utility that has, but there's definitely a signal in predicting this severity later in time from the metabolomic profiles. And then I want to draw your attention to E also. We also applied the same model, or a sub-model, 6 metabolites only, that is still pretty predictive in other cohorts that were published on COVID, and performed pretty well there as well on their respective definition of severity, which were different than ours. One was binarized and one was the 6 category WHO one. And in the one with the black bars, we were even better than their own model, actually, in their own data. So here another example now. This is from now an ICU cohort. These are different patients. These patients have ARDS, so that's acute respiratory distress syndrome. These are very, very sick patients. Death rates are between 25 and 50%. We'll see that in a minute in another slide. And here the question was, we had these patients with COVID-induced acute respiratory distress syndrome, but also patients with bacterial sepsis, ARDS, which is a different type of origin of the disease that looks clinically very similar when they're in the hospital beds. But it turns out, if you compare them multi-omically, and here we have metabolomic, lipidomic, and proteomic data, they're extremely different. So while the presentation to the doctor might be similar and they need ventilation and intensive care, it's a different disease, a completely different disease. And here we drew this map of all those changes that might be relevant for individualized treatment. And then on the right-hand side, draw your attention to B, the lower, the network. We then went inside of the COVID patients and complications they developed. One complication is thrombocytosis, is a massive accumulation of platelets, very bad side effect or comorbidity that occurs in these septic conditions. And what we saw when we generated this network and checked what correlates with this thrombocytosis, the interesting thing was that we saw multiple pathways, including TNF signaling and other signaling pathways, as well as extracellular matrix effects and cell adhesion, that all of which had been individually implicated in one way or another with this thrombocytosis phenotype. And here we were able to draw this now together and see that they actually seem to be very close in the pathway and talking to each other. This might be an angle for novel therapeutics. And now maybe the most mechanistically and clinically deep study that we've done on this, now going across different cohorts. There was a specific analysis, and I think you can't see the title, right? It's probably only in this room that you can't see the title. A specific analysis of vascular injury. Vascular injury is a very common phenotype in these types of conditions. So injury to the vascular system at the site of the disease, for example, in the lung. Also, again, very bad, very deadly if that occurs. So here we really try to now walk through different stages of the disease, from early disease to severe disease, through death, and then also recovery. So how this worked was like this. We had proteomics data, and when I say at-risk cohort, that is a specifically selected subset of that first cohort that I was talking about, the ones who are not in the ICU. We correlated proteomic profiles with death and with platelet counts. And this was the set of proteins that came up. And what you can see is here, the patients are in the columns, and on the right-hand side, there's a cluster of very severely sick people that have overall low abundances of these proteins that we found here. So it's a signature of about 16 proteins that if they're at low abundance, these people have poor outcomes. And now if we take the same proteins and go into the ICU cohort, right, so the ones that are now in an advanced stage of the disease, you can say that indeed they have severely increased levels of ANGP2, GPT2, which is a known marker of vascular injury. So indeed, if you have this protein signature that we already see early on in the disease, you have vascular injury. And way worse survival. So in this high vascular injury group, the survival is very poor. It's below 50% within days. And even in death, if in autopsy tissues, and 3 of these patients were actually from that ICU cohort, you see in the lung tissue signals of vascular injury as well as platelets. And since we had for 3 of those patients from the autopsies, we had the lifetime data, you can again see the worse the phenotypes are. Patient 3 is the worst here, the lower the original proteins were. And now, and this to me was the most fascinating part of the whole thing, after they recovered, so they came out of the ICU, we had another blood sample for some of them. Now the cohort gets smaller and smaller because follow-up is hard. So we had another sample of them, again found a group that had still this poor signature, and then a 12 months later, so there was a recall performance assessment, which is basically how well are you doing, are you in pain, do you have anxiety, can you live by yourself? And it turns out that those that even after recovery from the cohort, from the ICU, had a poor protein signature were not doing very well. They were alive, but they had problems 12 months after this whole thing happened. And I thought that was an interesting way of looking through sort of the trajectory through time, partially the same patients, and then partially through different patients of following up this disease with this particular problem, the vascular injury in COVID. Okay, so for the last few minutes, I'm going to focus on some of our cancer work. We also have a lot of work on now new on the more mechanistic side, trying to find combination drugs. I'm not going to show that today because that's mostly on cell lines and organoids and model systems. So I wanted to focus on the cohort related work today. So two pieces of work I want to show you. First, we have now metabolomics becomes one metabolite in this case. We have a study in follicular lymphoma. This is blood data before treatment. So this is no chemotherapy. This is really the raw signal at the time of diagnosis. Follicular lymphoma, you have to know, is a very slow disease. There's this watch and wait, so you don't even treat them. You just watch and wait. And what you can see here, and I don't know if you can all see the x-axis. So this one metabolite, formylmethionine, it's no score, it's just the metabolite itself already splits up the people by survival and over years. So we see here 3,000 days, so that's about a 10-year time frame where you can predict based on the blood concentration of that one metabolite in a slow-growing cancer that they will not do well with some accuracy. And the metabolite is absolutely fascinating. It's a mitochondrial metabolite. It has been associated with all-cause mortality in non-cancer studies, and together with the Cecchetti lab at Cornell, we're currently doing some wet lab experiments of what exactly is going on. Why does this metabolite occur? It has something to do with mitochondrial stress and some localization issues of proteins. And what was interesting, again, the Pan-Cohort aspect here is we were able to validate this in another cohort from the Mayo Clinic using a completely different technology, using an immunoassay, an ELISA, which is an interesting angle, right? So you can't always do mass spec on everything and everybody, but here this is cheap 96 well plates. You can buy it as a kit and it runs fast. And since this was only one metabolite, you can easily try this and measure this in more samples. Then I wanted to show that is our latest work together with Ed Resnick at Memorial Sloan Kettering, where we have been working on, and a lot of credit goes to him personally there, to build something like a TCGA for metabolomics, if you will. So we have all these different cancer types from different authors. There were emails to individual authors. Could you please share the data from your paper? And we put it all together, some of our own data in there as well, and have this catalog of about 988 samples with metabolomics, transcriptomics, some tumors, some normal tissue, that will soon be free, and then you can analyze whatever you want. As you probably know, clinical data is very hard to consolidate across different cancer types, but at least these molecular data will be all free. And just to give you an example of what you can see, and again, you can do a million things on this, that's just an angle that we looked at. We checked which metabolites correlate with which gene expression profiles across the different cancers, and the surprising finding was that let's say it's not what you might think it is. It's not that, you might think in a tissue transcript metabolite, it's something like enzymes and transporters, but it actually isn't. It's mostly distal relationships of higher level regulation, and we're still looking into what it is. It looks like there's a lot of immune signal in there. So you have these hub metabolites that correlate with a lot of different transcripts, and that seems to have to do with immune processes. We're still currently working this out in the paper. Last slide of content, and then I want to have a quick thank you slide. So a new passion project that arose after we talked with our colleagues at the veterinary medicine hospital in Ithaca is comparative oncology. So you compare cancers across species, not induced cancer, but spontaneous cancer, the actual disease, not a lab-made version of it. And the doc apparently, which I also learned recently, is a great model for this. So we already had for a while diffuse large B-cell lymphoma human samples. We've had them for almost 10 years. And now we recruited with them, and they're great nurses at the hospital, doc patients, also diffuse large B-cell lymphoma, and ran a metabolomics analysis on it. I'm just showing some very preliminary data. This data came in weeks ago. You see a lot of differences between the normals and the cases, and with them comparing this with humans. All right, I want to thank everyone. Also want to say briefly, we're hiring. If you know bioinformatics postdocs, send them my way, either academia or startup. We're looking on both sides. So if you know anyone, please send them my way. And thanks.